Okay, here we are continuing from the first video. We're here in visual cortex. We're in the primary visual cortex, and we're looking at how different types of cells seem to be stimulated by different visual features, particular orientations of lines or particular colors. And we're doing this as a part of tracking the two visual pathways, the how pathway and the what pathway, as they travel through these early visual cortical areas. And we can see how even at these early levels, the information is being kept separate between the two pathways. So here's the good example. So here's the lateral geniculate nucleus sending the magnocellular and parvocellular layers to different layers of the primary visual cortex, which synapse to different other layers of V1 and then on to different sections of V2 and through on to different parts of the visual cortex and other parts of the brain as a whole. So here's those blobs in V1. Here's the incoming information from color going through the blobs into the thin stripes and into V4. However, the motor cortex has this direct route between primary visual cortex that bypasses V2, goes directly to the motor cortex, which we'll see again in this next slide. This is a slide from the textbook showing the same image we had of the thalamus. And then the incoming information from the thalamus to V1 synapsing on different layers of V1, VC, 4CA versus 4C beta. And those different pathways then ultimately lead to different connections into V2 and onto the motor cortex. So you can see that there's a direct link between the thalamus, V1, and the motor cortex, which bypasses V2 altogether. That's part of this where or how pathway that allows us to interact with the world. These are some stains of V2 and MT showing some of the stereotypical features of them, and here's the blobs of V1 you can also see. So here is that image from 2 slides ago, but fleshed out even further anterior into the brain, showing the ultimate direction that these pathways take. So they start off as separate visual pathways, the M and the P pathway, and they ultimately end up leading to these very two different broad brain pathways, the parietal pathway and the temporal pathway, or dorsal and ventral, or how and what. And so the type of information that gets transferred into each of these pathways obviously is relevant for what their function is going to be. And this next slide shows that quite well. You can see the particular types of features that neurons in each of these stages of the pathway process. So here you can follow the color information from those blobs and inner blobs into the thin stripes of V2, onto V4, and then on to the visual processing, which we'll talk about in the rest of this section in the temporal lobes. On the other hand, something that we'll cover after the first midterm is this pathway up in that seems to process spatial orientation of objects, how we need to reach out and grab them, how we need to move our eyes toward them, and also, as we'll see, how we need to attend to certain parts of them and ignore other parts. So the evidence for all of this is now over 60 years old almost, and it comes from studies of monkeys and human lesions as well. The studies of monkeys were done at the University of Wisconsin, the same university as the author of our textbook, and they were done using an apparatus like this. You have a monkey that you put in a cage, you have an experimenter outside of the cage. The experimenter can manipulate what's being presented to the monkey on this tray, and included in the tray and maybe under a certain one of these blocks is some food that will reward the monkey, and they'll try to teach the monkey certain behaviors by rewarding and punishing its actions. And so one example that we're going to see today is they're going to see how well a monkey can remember and learn which of two locations had a food reward. We're also going to compare that to situations where monkeys had to learn to identify objects that always had food under them. And we'll see how different manipulations you could do inside this Wisconsin General Test apparatus could allow us to tease apart these ideas. So here's an example of two of the tasks that are apparently quite similar, but in fact have very subtle differences that lead them to utilize entirely different parts of our brain when we do them. So the landmark task on the left is like finding out that your friend is waiting for you under the CN Tower in Toronto, and you can see the CN Tower, and you know that there's a food reward with your friend waiting for you, and you just have to get to the location adjacent to the CN Tower. So the monkeys are learning to relate the food to the location with respect to a nearby landmark. On the other hand, this task has two objects that can be in any position in the world, and the monkey always has to learn that the food is going to be under one of them, say the pyramid and not the cube. Okay, so these tasks are similar in that the behavior the monkey needs to do is very similar. They need to just reach out into these different stimuli and touch them to uncover the food underneath the two locations. However, we'll see that lesions to monkeys when they're attempting to do this task can drastically interfere with the ability to do one task but not the others, depending on where the lesion is. So the experimenters either lesioned the parietal lobe of a monkey or they lesioned the temporal lobe. And what they do is they'll get the monkey to learn where the food is, and then they'll switch and put the food on another location. So those are these reversals. And in this case, they had the monkey guess where the food was, and then they teach it that it's under one of these things, and then all of a sudden they'll switch and reverse it. And so what you can see here in an unoperated monkey is that originally they make a bunch of errors as they learn which location has the food, and then they eventually learn, and then they reverse it, and they immediately get worse because they check back in the old location. But after the next reversal and the next and the next and the next and the next and the next, they get better and better and better. They make fewer and fewer errors after the reversals. So in theory, they're learning to associate these landmarks with particular locations in the world where they can get food. If you damage the temporal or the frontal lobe, their behavior stays quite similar. However, the researchers were surprised to find that when you damage the parietal lobe, this area in the how pathway that we've just observed receives a bunch of input from the magnocellular parts of the visual system. When we lesion the parietal lobe, the monkeys do bad during the first reversal, but then they fail to get better over subsequent reversals. But one issue is that the monkeys may just have had maybe a hearing problem or a sight problem, and so they could just be, maybe the parietal lobe is important for some behavior that's not necessarily identifying objects and where they are. So the monkeys in a control condition had a lesion instead of their temporal lobe. And so now instead of their frontal and parietal lobe, their temporal lobe is lesioned. And they do this different task. So notice how monkeys with temporal lobe lesions could do this task just fine, but when you get them to do this other task in which now they have to identify which object the food is under and remember that over the course of the trial, and then it reverses and to be under the other object, you can see with a frontal or parietal lesion, monkeys are quite good. They don't need to improve very much. With a temporal lesion, the monkey does very poorly on this, and only after a great number of repeated exposure to this task do they begin to get better. So we call this a double dissociation, that these two brain areas doubly dissociate with these two behaviors. This brain area affects this behavior and not this one. Damage to this brain area affects this behavior and not this one. So this is really strong evidence of the involvement of these two brain areas in these two different types of tasks. So, and this is where the original kind of findings of the how pathway or the where pathway versus the what pathway originated, is in these kind of studies from the 1960s and 70s, I think. So here's an example of the pathway in the macaque showing the what pathway, which we're going to cover for the rest of this topic in the textbook, and then this how pathway, which we'll cover in chapter 8, I believe it is. So what we didn't cover in the textbook, and what you might learn about in 377, is that there's patient work around the same time where patients came in with lesions to specific parts of their brain, and they were able to identify patients that had damage on certain tasks but not others, in much the same way of these monkeys. So here in this case, we have this patient DF who can, when she's asked to reach out and put a letter into a mail slit, can very reliably reach out and place the letter at the particular orientation that this slit is oriented. So you could orient it this way and she'll always be quite good. However, when you just ask the patient to instead indicate, without posting the letter, but just to say with a keyboard which direction the orientation is, now they have a great difficulty, and she indicates basically randomly which orientation the mail slit is. So it seems that when she can physically interact with the world and reach out and grab something, she's able to do it, but when she actually has to observe the world in front of her and report it, she isn't. And so I hope you can guess which of the two brain areas from this study were DF's damage in. DF had damage that impaired her ability to recognize objects, but didn't impair her ability to interact with them in the world. So she likely had damage to her temporal lobe. And the researchers that did these studies found similar patients with the opposite effect, where they would be able to identify visually which direction the slit was, but couldn't reach out and post it correctly. So for the rest of this lecture, starting today and on Thursday, we're going to cover the rest of this what pathway through the visual system. What do all the neurons on the underside of the temporal lobe do as they transfer information from these low and simple signals about lines and colors and shapes up to these more complicated brain areas? What we'll see is that a couple of things change as we move up to the higher levels of the brain. One is that the size of the receptive field gets bigger. So as more and more and more of these low-level signals combine into higher and higher levels, they represent a bigger and bigger section of the visual world. Also, another thing is that the neurons, if you time when they fire with respect to when a stimulus comes on the screen, they take longer and longer to fire because they have to pass through more and more brain areas before they get their information. Finally, another thing that they share in common as we move up the hierarchy is that more and more complex features are represented. So whereas simple dots and lines are represented in V1, in the highest levels of the temporal lobe, we have neurons that respond to particular shapes of particular objects, irrespective of their position in the visual field or their size or direction. Let me check on the time for this lecture. Looks like we have another minute to finish, which is good because we have about one more slide here. So we'll cover this again on Thursday, but here's a crazy view of the visual hierarchy showing how information funnels from these low levels up to these highest levels into the hippocampus. One thing to mention is that there isn't clear separation between the what and the how pathway. There's actually a lot of crosstalk between them. Here's examples of the receptive fields of a what neuron and a how neuron. So a neuron in the temporal lobe versus a neuron in the parietal lobe. Neurons in the temporal lobe have receptive fields of fixation because they pay attention to what it is that you're looking directly at. And neurons generally in the parietal lobe have receptive fields contralateral to where they are in the brain, and usually in the periphery because they're more involved in detecting things like motion. And with the last 20 seconds, I'll give you a preview of the fact that there are neurons in the temporal lobe that respond to hands. So there's body part sensitive neurons, and we'll see there's also face sensitive neurons, and we'll learn about how they might come to have this signal.